Okay, so let's start off by just reviewing a little bit of what we talked about in the beginning part of the notes, and then we'll go ahead and we'll talk a little bit more about enzymes and that final organic compound group, nucleic acids. So after taking the notes last week and doing the lab, you should be able to look at these pictures and tell me if you're seeing lipids, carbohydrates, or proteins. So you should think about the models that you built and find the one that matches most closely to the picture. So let's start with carbohydrates. The carbohydrate model that you guys built, first you built a monosaccharide, and then you linked two monosaccharides together in a condensation reaction to make a disaccharide. When you look at these pictures, you should recognize that this right here is the closest picture there is to another carbohydrate or a monosaccharide. This is a monomer. I look at it and I see that ring-shaped structure that you built, roughly the hexagon in the middle of the molecule. Anytime you see that, you know it's a carbohydrate. And then this could bond with another monosaccharide to make a disaccharide and keep bonding to make a polysaccharide. The next thing you built was a lipid. Looking at the remaining pictures, you should be zoning in on this picture right here as your lipid. Now this picture it's a little bit harder to see. That lipid kind of probably looked like one big tangled molecule. But if you very carefully went into the lipid that you built, you should have seen that E-shaped structure. The glycerol, which we sometimes call the backbone, and then the three fatty acid tails coming off of it. So the lipid you should recognize is that E-shaped structure. This right here is actually also part of a lipid. It's one of the fatty acid tails right here. And then finally, this last molecule is obviously your protein. Now the dead giveaway for this being the protein is the fact that it says peptide bond. And you know that peptide bonds are the special name we give to the condensation reaction of proteins. The condensation reaction that occurs here is no different than what would occur in a carbohydrate or in a lipid. There was a condensation reaction here, here, and here. As those fatty acids linked into the glycerol, each time it was a condensation reaction and a water was lost. But proteins are so important to our bodies that we give that bond a special name. We call it a peptide bond. What you should be able to see here is that this is actually a dipeptide or a dimer. There's one amino acid right here and another amino acid right here, and we built these in the lab. Has its central carbon, has the R group coming off, has an H coming off, has the carboxyl group, and the amino group. You should be able to diagram the condensation reaction that happened right here. Let's do that together. You should be drawing this in your notes. You should be able to tell me what was lost from this side and what was lost from this side in the condensation reaction. So we know that right here there should have been a hydrogen. So that hydrogen was lost. And we know that over here there should have been an oxygen and a hydrogen, a hydroxyl group. So that hydroxyl group was lost. If I put this OH and this H together, that will make H2O. It left a bonding site open so the two amino acids could bond together, peptide bond, and you had your water formed. That was the condensation reaction. So it should be very clear that the carboxyl group lost this hydroxyl and the amino group lost this hydrogen. Practice looking at these before the test. Look for those patterns. Look for the ring in the carbohydrate, the E-shaped structure in the lipid, and then look for the peptide bond in between the amino acids. We want to talk a little bit more about proteins before we move on to the final organic compound group. And we want to talk about the role of proteins as enzymes. We said back in our notes, if you look at your graphic organizer, that one of the major important functions of proteins is that it is an enzyme. What does that mean? Well, we should remember, we talked about in chapter 2, you talked about in past science classes, that enzymes are molecules that speed up chemical reactions, let them happen faster, or lower the activation energy. So remember we talked about how in the chemistry class, if you want to have a reaction happen faster, you just heat it up, you put the Bunsen burner on it. But we can't do that in our cells. I can't put Bunsen burners inside my cells to digest my food. So instead, I use enzymes. Enzymes are little three-dimensional structures that inside my cells can speed up chemical reactions, or lower the activation energy without the need for heating the reaction up. We talked about the fact that there's two different structural types of proteins. There was fibrous proteins and globular proteins, and you should be able to guess that these enzymes are going to be globular proteins. These aren't going to be long, thin strands like spider webs. These are going to be little blobs floating around in your cells. Now, enzymes, you don't need to memorize the names of any enzymes, but on a test, you should easily be able to pick out enzymes out of a list because they usually end in the same three letters, ASE. So you don't need to memorize these three enzyme names: maltase, amylase, carbonic anhydrase. But if I put them on a test, you should know, oh, those are enzymes, those are proteins. Another thing you should know about enzymes is that their function can be regulated by temperature and pH. And well, why does that matter? That matters because your body can have different temperatures. And your body certainly right now has different pHs. The pH of your stomach is very acidic, while the pH of your intestines is not. So the fact that enzymes only work at certain temperatures and certain pHs, that's what this means. They can be regulated. Enzymes only work at specific temperatures and pHs. That gives your body a little control over how the enzymes work. So let's graph this. Let's say we're looking at pH, and over here we're looking at the effectiveness of the enzyme. So how much is the enzyme working? The effectiveness. So let's say we would graph an enzyme that works in your stomach. So stomach pH is pretty low. So on that pH scale of 0 to 14, that enzyme's really going to work only down here in the area of the acidic pH, and then it's going to drop off again and probably not work in basic. So their enzyme function's regulated. It only works at certain temperatures and certain pHs. And one more thing you should know about these little globular enzymes floating on your cells is that they can be used over and over and over again. On the next slide, I'm going to show you kind of what they look like as they're working. I want you to have a basic understanding of how the enzyme works, and there's some vocab that goes with this. So just take a look at this animation that's happening on the screen. You should see that little black enzyme clicking into the red substrate and then breaking apart. And this is going to happen over and over and over again. That red enzyme's used again and again and again. And that substrate clicks into it and then breaks down. So essentially what you're seeing right there in the animation and in the diagram above is a hydrolysis reaction. Maybe that black substrate is a disaccharide, and it's being broken down into two monosaccharides by the enzyme. Let's make up some numbers just to put this in perspective. Remember, enzymes are lowering our activation energy. Maybe that disaccharide only breaks down if it reaches 150 degrees. Well, you just ate lunch or you just ate breakfast, and you need that disaccharide to be breaking down in your stomach at your body temperature. So the enzyme lowers the activation energy. I don't have to put a Bunsen burner inside of you to heat it up, and it allows it to break down at a lower temperature. Okay. Now, what you should also notice is that enzyme and the substrate are clicking together just like puzzle pieces. Substrates are very specific. They only work with one enzyme, and we call that the lock and key model. That's why I have that picture of the lock and key. Just like your house key only works on your house and it doesn't work on your neighbor's house, enzymes only work with one substrate. So if this enzyme right here is working with, come on, pointer. If this enzyme right here is only working, let's say, with the disaccharide, like I said, let's say this is a disaccharide clicking in here, it would not fit with the dipeptide. Maybe the dipeptide is square in shape, and it would have to have its own enzyme that it could fit with. Now, there is some vocab in here that we've been using. When the organic compound is fitting in with the enzyme, we call it a substrate. So the first set of vocab words, substrate and enzyme. When they click together, we call it the enzyme-substrate complex. And that site right there, where it fits in perfectly like a puzzle piece, that's called the active site. So it's just a little bit of vocab here to describe what's going on. Again, both the animation and this picture are